Good morning, dear dignitaries and all the delegates. Welcome to the day one of 1 week online national FDP, organized by Shri Vishnu College of Pharmacy in association with Crystal Growth and Nanoscience Research Center, Department of Physics, Government College, Rajahmundry, and with the IPA Educational Division. And I would like to, dear dignitaries and all the delegates, welcome to the day one of 1 week online national FDP, organized by Shri Vishnu College of Pharmacy in association with Crystal Growth and Nanoscience Research Center, Department of Physics, Government College, Rajahmundry, and with the IPA Educational Division. And I would like to, dear dignitaries and all the delegates, welcome to the day one of 1 week online national FDP, organized by Shri Vishnu College of Pharmacy in association with Crystal Growth and Nanoscience Research Center, Department of Physics, Government College, Rajahmundry, and with the IPA Educational Division. And I would like to, we are happy to inform that we are having very good and eminent speakers from various national institutes, NIPER, National Institute of Technology, Warangal, as well as from various government organizations, government colleges like Untitled College, Pharmaceutical Sciences, Krishna University, and private universities, JSS University, and SRM University.
Sir, in Zoom, please, everyone, please mute, sir. Sir, everyone in the Zoom, please mute, sir.
Krishna University, and private universities, JSS University, and SRM University.
Sir, in Zoom, please, everyone, please mute, sir. Sir, everyone in the Zoom, please mute, sir. And on this event, I would like to introduce regarding our director, sir, who is going to deliver a keynote address. Dr. Kumar, V.S. Nemoni, Director, SVCP, from since April 2018. He holds his B.Pharm, M.Pharm in Pharmacology degree from Andhra University, Visakhapatnam. He has completed his PhD degree under the guidance of ex-professor and Director P. Ramarao Naipar Mohali. Subsequently, he was associated with Professor Jeffrey S. Mughil, McGill University, Montreal, Canada, for his postdoctoral fellowship for 3 years. Dr. Kumar V.S. Nemoni has associated with the leading research centers in India, namely Dr. Reddy's Research Foundation, Hyderabad, Piramal Life Sciences Limited, formerly Nicholas Piramal Research Center, Mumbai, and Lupin Limited, Pune. He has over 14 years of drug discovery and drug research, development research expertise and managerial experience. Now I request our director, sir, Kumar sir, to give a keynote address to the gathering.
Hello everyone, and welcome to today's national webinar on nanotechnology in healthcare. First of all, I am overwhelmed with the overwhelming response from the all the participants. The number of registrations have crossed more than 1,000, and more than 17 states have participated for this webinar, and more than 175 colleges have registered for it. I think the credit goes to the organizers and all the panelists that have agreed to give this talk on this specific topic, that is nanotechnology in healthcare. On behalf of the management, our chairman sir, Shri K.V. Vishnu Raj Garu, our vice chairman, Ravichandran Rajgopalan Garu, and the principal sir, Dr. K. Prasad Garu, and all my staff, I welcome each one of you to this webinar. And I also like to thank and welcome all the principals and the senior staff who are highly supporting to this event. I think all of you know more about nanotechnology in the coming days. I think experts panel will talk about nanotechnology and its impact on the healthcare. I'd like to thank all the panelists who have readily agreed to talk on this webinar. Especially, I like to thank my teacher, my professor, my guru, my mentor, and scientific advisory board member of SVCP, and more importantly, governing body member of this college. That is none other than our inaugural speaker. That's our professor, our beloved professor and highly respected professor, Professor K.V. Ramamurthy Garu, Principal, Andhra University College of Pharmaceutical Sciences. And many of you know him, and to his credit, he has a number of PhDs awarded and excellent publications. I think this is the best way to start a webinar from a college setting. Thanks a lot, sir, for readily accepting and honoring us in giving this inaugural lecture. I'm sure with this webinar series, all of you will be excited with the science as a career, and especially in nanotechnology. And we are going to organize this webinar series from each department. And the next one is on drug discovery and development, and most of the speakers from international expertise. I'm sure you all love this and make best use of it. I also request and suggest to all of you to participate actively in this webinar and seek the guidance from the expert panel available, and also suggest to your friends to join this movement of educating the pharmacy fraternity. So with this, I once again welcome each of the delegate who has joined this webinar and wish that you have a wonderful time. And please excuse us for the technical lags, and we hope with your feedback and suggestions, we'll improve day by day and make this event much more successful. So with this, I thank you all once again and welcome to this webinar series. Ashok, over to you. Ashok, you need to unmute.
Now I am delighted to thank you for your keynote, Director Sir. Now I am delighted to introduce our today's guest speaker. He is of renowned person and face of the pharmaceutics throughout the renowned throughout not only India. He is having a worldwide recognition. And today's our guest speaker, Dr. K.V. Ramanamurthy Garu, Principal of Andhra University College of Pharmaceutical Sciences. And today he is going to deliver a guest talk on nano drug delivery systems, current trends and future perspectives. I would like to give a brief biodata information regarding the sir. Sir completed his bachelor's, master's, and PhD from the Andhra University College of Pharmaceutical Sciences. Later, he rendered his services as lecturer, associate professor, senior professor in the Department of Pharmaceutics in the same Andhra University. Currently, sir is the principal for the Andhra University College of Pharmaceutical Sciences. Added to his reputation, he is having the enormous number of international and national journal publications with high impact factors, and he is guiding more than 50 PhD scholars. Now I am giving this session to the Ramanamurthy sir. Over to Ramanamurthy sir.
Yeah. Very good morning to all my friends, students, some of them who are listening to me. First of all, I would like to thank Vishnu College of Pharmacy for giving me this excellent opportunity to share my thoughts on nanotechnology. And I am very much thankful to Kumar for the nice words he has spoken about me, which has shown his affection towards the teacher. I am really thankful to him. Today, I think all of you could see my presentation, okay?
Not yet, sir. Share screen.
Share screen, not yet, huh? 1 minute.
Yeah, not yet, not yet.
Now I think, is it okay?
Yes, sir, it's coming.
Yeah. Today I would like to discuss some concepts, what I would like to share with the audience related to nano drug delivery systems, current trends and future perspectives. I would like to cover about what are nano drug delivery systems, what are the challenges in their developments, what regulatory considerations one need to follow, and what are the future perspectives that can be possible for this. So these nanopharmaceuticals can be manufactured by using physical, chemical, and biological approaches. And as far as US FDA is concerned, they have allowed the commercialization of about 100 nanomedicine applications and products. And what do you mean by a nano formulation or a nanopharmaceutical? According to the European Commission recommendation, nanomaterial refers to a product that is obtained either natural or occurred through incident or by manufacturing, which comprises the material either in an unbound state or in an aggregate, where one or more external dimensions is in the size range of 1 to 100 nanometers, where 50% of the 50 or more than 50% of the particles according to the number size distribution. So the definition is very critical, and people may think the different approaches, but as per European Commission, one dimension is sufficient enough to have a nano size range in the 1 to 100 nanometers, where the number size distribution is only considered for the nano material classification. What is the need for the design of these nanostructures? In this particular slide, I tried to give these sizes of different materials, but what we encounter in our daily related to, where the water molecule is having a very small size of an angstrom, approximately 1 nanometer, protein molecules are 5 nanometers, and from 10 to 100 nanometers, you have different particles like body viruses, etc. And bacteria around micrometer, and human ranging from 10 micrometers onwards. So nanostructures are highly advantageous for different reasons. One is their size range comparable with the biovacuum. They have large surface area such that the volume is very low and surface area is very high. And if you take a silicon nanowire, it has a small size range. Are you able to listen to me, Kumar?
Yes, sir, we are getting you.
Clear.
Okay. 10 nanometers, if it can have an accommodation for that. Based on this assumption, if we take one square cross section of a nano wire, it accommodates 10 atoms associated with that. But if you just increase the size to a macroscopic level, where a 10 mm wire with a cross section can have only out of 10,000 atoms, only one can be associated with the surface of the wire. So this clearly indicates the capability of association of the nanostructures with the different biological surfaces, and that made and created a lot of interest for the development of these nanostructures. What are different nanostructures we have? Already we know these structures to a great extent, like nanocrystals, polymeric nanoparticles, liposomes, micelles, dendrimers, mesoporous silica nanoparticles, inorganic nanoparticles, carbon nanotubes. So what are these nanocrystals? These are crystalline particles produced by different methods like parallel milling, high pressure homogenization, precipitation process, etc. What are the advantages? They have improved dissolution rates, they have improved saturation and very safe for the immune and able for intravenous injection without any problem, and they give viable to high dissolution solution rates. Next comes the polymeric nanoparticles. They can be pronounced polymer coating, and the advantages they can be administered by different routes like infusion, injection, or oral. They can have like gene delivery, cancer therapy, or brain barrier. They can carry many aids in the small, and they have very good thermodynamic stability, and they can penetrate with cells, tissue, the nanoparticles. And the polymeric nanoparticles are marketed in US and other countries. Next comes the liposomes, a very old topic which we already studied number of times. Just I don't want to waste the time by highlighting these things, but still they are very important for drug delivery, and they are formed from lipid bilayers, and they may be divided into unilamellar or multilamellar, and they can dissolve both water soluble or lipid soluble. That means they can entrap, and they can have both the drugs simultaneously in the same molecule. And they can be classified as conventional liposomes, pegylated liposomes, ligand-targeted liposomes, and multifunctional liposomes for various applications. The advantages of liposomes are passive targeting, high efficient cargo delivery, that means more entrapment, drug-reduced cargo toxicity, that means the toxicity is comparatively very less. And these are the marketed formulations of liposomes. Next comes micellae, and they are spherical amphiphilic copolymer nanoparticles formed by supermolecular assembly, and they have the core shell structure with hydrophobic inside and hydrophilic outside. So the diblock copolymers are joined together for the formation of micellae, and these micellae are going to be lyophilized, and when they are hydrated, we are going to have the formation of these micellae. And the advantage is they have very high loading capacity and very good stability in the blood, and they have prolonged circulation time and less number of side effects, and they can protect the internal drugs from degradation because their drugs are entrapped in the core-like structure where degradation is prevented to a great extent. Next comes dendrimers, These are the synthetic tree-shaped macromolecules having a 3D structure with branches extended from central molecule like second generation, third generation, fourth generation, and we can modulate the size according to the generation number. The advantages of the dendrimer is they can have different attachments for different branches of this dendrimer, wherein we can have a bioactive molecular drug, we can have the drug, or we can have the imaging agent, or some other fluorescent tags, etc. And we can have a defined molecular weight for this dendrimer as per our tailor-made requirements. They can have uniform in shape, and host-guest envelopment properties it can have, and it is having very low polydispersion. That means we can obtain a very uniform size. Mesoporous silica nanoparticles are another class of nano drug delivery systems. They are silica particles in nano size range with large surface area and a solid structure. They have also having high loading efficiency and good protection ability by keeping the cargo molecules inside the pores. That means the drug will be protected from degradation, and they have controlled release ability. Inorganic nanoparticles is another class wherein we will be using the metallic elements like silver, gold, copper, zinc, magnesium, etc. And they can also be made for a customized size. They have large surface to volume ratio, and we have surface plasmon resonance and different noble optical properties. They are easy to synthesize, and they have different surface functionalization properties. They have very low toxic ability, and they can be inert in nature. So we have already seen so many inorganic nanoparticles for their applications in the earlier literature. Last one is carbon nanotubes. They are needle-like carriers. They can easily diffuse into the cell membrane by perforation, and they have they are classified into single-walled as well as multi-walled, having different physical properties like Young's modulus and electrical conductivity, etc. And they have prolonged circulation time, cell membrane permeability, and high aspect ratio, allowing high drug loading. So as far as our research group is concerned, we have done some work. I don't say it is a considerable work, but still we tried to put our mark in the nano drug delivery systems, starting with 2013-14 onwards, where we published some good publications according to my perception in good reputed journals. And this is the first work what we attempted for the nano formulations by preparing a dendrimer of second generation, third generation, fourth generation dendrimers were prepared, and they are thylated with the sulfur groups. And we tried to evaluate the mucoadhesion properties of these thylated dendrimers by using acyclovir as a model drug. And we proved that the drug release is extended for 50 hours almost, and it is able to have very good mucoadhesion compared to the pure drug or dendrimer loaded drug molecules. The second work is the formulation characterization ex vivo studies of terbinafine hydrochloride liposomes for cutaneous delivery. When we started this work, nobody attempted the cutaneous delivery of these liposomes by incorporating them into the gel. But we have two goals for doing this work. One is the terbinafine is given orally for treating the nail and other infections, but we wanted to deliver directly to the infected part of the nail such that we can have more therapeutic effect. The second one is we are working on the natural polymers like gum karaya, hupu gum, etc. in our laboratory. So we wanted to test the efficacy of this gum karaya gel for the transportation of this particular to the cutaneous delivery. And we studied the different parameters, and we found that the discussion showed prolonged drug retention at the cutaneous part, as well as the ex vivo permeation studies also proved that it can go for a prolonged time. We completed one more good work wherein a ficetine molecule was tested for the preparation of the polymeric nanoparticles, which is one of the very good in collaboration with IIT Hyderabad and along with Ramakrishna Sisla. And Physin is a drug which is having a very poor solubility, and we try to improve its solubility. And by preparing the H-beta CD complex Physin, improve the solubility of the drug from 5 approximately 860 micrograms per ml. And by using this drug complex, we prepared the polymeric nanoparticles and evaluated the polymeric nanoparticles for various characteristics like the drug release, the cytotoxicity studies, and cellular uptake studies, and other studies. And we have done even the in vivo studies in the mice. And the results clearly confirmed the applicability of this Physin through oral route, wherein there are no earlier reports of the administration of this Physin through oral route, and we are successful. And we improved the bioavailability of this particular Physin when administered through oral route by about 13 times, and the Cmax by about 8.5 times, which is a significant contribution. And we wanted to test the antibacterial activity of these metallic nanoparticles as well as anti-cancer activity of these metallic nanoparticles. And majority of these metallic nanoparticles are by using very corrosive approaches, by using the hard chemicals for causing the reduction of these metals for formation of the nanoparticles. We wanted to prepare the nanoparticles by using the green synthesis approach, wherein we tried the ananas squamosa leaf extract, which is going to be used as a reducing agent. So we prepared the ananas squamosa leaf extract of different extracts, and finally we optimized all these extracts, and we found that phenols are more in concentration in the aqueous extract, approximately 150 mg per ml, 150 micrograms per ml of phenol content in the aqueous extract, which is highest. And we contemplated that this can be used as a reducing agent for preparation of the metallic nanoparticles. So we tried to prepare the silver, zinc, and copper metallic nanoparticles by using this ananas squamosa leaf extract, and we are able to synthesize and characterize these 3 different metallic nanoparticles, and we evaluated them for various applications. We compared the anti-cancer activity of the silver nanoparticles both on the cancer cell lines or healthy cell lines, HEK cell lines and HeLa cell lines, and we found that up to a concentration of 200 micrograms, they are safe on the healthy cells, and at a very low concentration of approximately 25 micrograms per ml, they are very effective for the cancer cells. So we concluded that they are not causing any damage to the healthy cells, but they are able to target the cancer cells for causing the anti-cancer activity. Similarly, we qualitatively tested the in vivo wound healing activity of these particular nanoparticles, and we found that they are able to give very good wound healing activity in conjunction with neomycin when it is applied. That means it is showing a synergistic activity when it is applied to the wounds of a rat. So this was actually a collaborative research work in collaboration with the Vishnu College of Pharmacy, because majority of the work was carried out at that particular Vishnu College of Pharmacy. And myself, along with P.V. Swami, we are able to produce this PhD as well as the different applications. Second one is the zinc nanoparticles. We also got the similar results with the zinc nanoparticles, but however, compared to silver, copper, and zinc nanoparticles, the concentration of zinc nanoparticles is slightly higher. For copper and silver, we are able to get 25 micrograms per ml, whereas with this, it is 50 micrograms per ml. Why these nanoparticles are showing a lot of interest to the doctors? Because majority of the academic and industrial research laboratories are constantly making advances in nanomedicine research. The success in the nanomedicine research led to the development of different nano drug delivery systems for the successful treatment of diseases like cancer. And majority of these nanoparticle drug delivery systems containing the drugs like Doxil, Abraxane, etc. have received the FDA approval due to their better efficacy and less side effects in comparison with the pure drug. due to EPR effect related to nanoformulation. However, the other side of the coin is a formulation by Bind 014, an anti-cancer nanomedicine made of docetaxel coated targeted ligand, which is believed to be safer and efficient than docetaxel, found success in small clinical trials. So people thought it is going to be a big bang, but when it is gone for the larger sized clinical trials, it failed miserably and the trials were shelved. So a warning that indicated that nanomedicines can be prepared at laboratory level very successfully, but when they are being taken for the large scale manufacture, one should be more careful. So we need to bridge the gap that is going to be there between the laboratory product and to that of the commercial product, having developed different nanoparticles for the mankind. But it is a diminishing attribute that may disappoint the researchers and industries. But once again, I feel it is not a diminishing attribute, but it may be a what we call is learning success from the failures what are faced by us. So what are the challenges we need to address while developing these nanopharmaceuticals? So first one is the physicochemical characterization. Second one is biocompatibility and toxicology. Third one is pharmacokinetics and dynamics establishments. And fourth one is process control and scale. So what are the physicochemical characterization studies one need to carry out? So the starting point of the characterization are the particle size, shape, size distribution, aggregation and agglomeration, crystal structure, surface chemistry, charge, etc., etc. And we have different techniques for doing this physicochemical characterization. And broadly, we can group them into counting, ensemble, separation, and other characteristics. So what are these counting methods? They are the measurements for the individualized particles, sizes, visualization, and their morphological examination by using different microscopic methods. So we can use TEM, high resolution TEM, scanning electron microscopy, cryo-SEM, atomic force microscopy, particle tracking analysis are some of the methods. So we can classify these nanomaterials into three different parts, like nanomaterials having 3 dimensions below 100 nanometers, and examples are quantum dot or hollow sphere. It may have only 2 dimensions below 100 nanometers, like a fiber or a wire, or it may have only 1 dimension below 100 nanometers. It can be a film or a coating of a multi-layer. So majority of the methods what are listed here have a disadvantage, wherein these are operated under high vacuum and there may be a chance of dehydration of these nanoparticles or nanopharmaceuticals. So cryo-SEM is the only one which we can use it more confidently for preventing the dehydration under high vacuum conditions. The ensemble methods involve the intensive particle size measurements. So here we are not going to count the number of particles by but we can measure the intensity of a specific particle size distribution, and we may be using a dynamic light scattering or small angle X-ray scattering or X-ray diffraction. So DLS and quasi-elastic light scattering techniques are based on the Brownian motion of the sample, and XRD is a good technique to obtain the information about the chemical composition, crystal structure, and physical properties. Separation methods are also very important because it is always advisable to obtain a monodispersed fraction rather than a multi-dispersed fraction of the nanomaterials. So we need to have different separation methods like field flow fractionation, analytical centrifugation, and differential electrical mobility analysis. These are some of the techniques that are used to separate these nanomaterials into a monodispersed fractions. Integral methods, they use only for measurement of the integral property of the particle, and they are more used in this area. We use the BET adsorption isotherms for evaluating the nanomaterials. And we also use electrophoretic light scatter technique for determining the zeta potential which is a parameter related to the overall charge of a particle that occurs in a particular medium, and it is also going to correlate with respect to stability of these nanoparticles. The problem of biocompatibility and nanotoxicology should be properly addressed. So, if we cannot establish the biocompatibility and nanotoxicology of these nanomaterials, even though the system is highly considered to be advantageous and successful, they may have the toxicological concerns. So we have to address these problems, how they are penetrating into the cells and tissues, and how the biodistribution is going to occur, how the degradation of this nanomaterial is going to occur, and finally how they are going to be excreted are also to be established, because majority of these nanomaterials exhibit different ADME pathways compared to the parent drugs. Hence, we have to be very, very careful in establishing all these parameters. And doses. Sometimes when these nanomaterials are prepared with some doses, they may have different application, different effects on the biological system compared to the parent drug. That also to be addressed properly. So we have to have proper preclinical assessment of these nanomaterials, and a thorough biocompatibility testing program is also to be done by using both in vivo studies, which are to be complemented further by developing proper in vitro assays, because we cannot do always the in vivo studies every time. Hence, initially we do the in vivo studies for establishing all these biocompatibility and nanotoxicology, but once they are established fully, we need to develop some suitable in vitro assays to prove the safety of the batches of the nanomaterials that are being prepared in the industry. So, for evaluating the nanotoxicological effects of particular nanosystems, Keck and Muller proposed a nanotoxicological classification system like our BCS classification system, and that is going to rank these nanomaterials into 4 different classes. So class 1 is the size above 100 nanometers and they are biodegradable completely. So it is a very safe system for the nanotoxicological part of view. And class 2 system is size above 100 nanometers but non-biodegradable. That means we need to have some caution in its usage. And class 3, they are size below 100 nanometers and biodegradable, but still we need to have some caution because they may be distributed into different tissues and they may cause some other damages to the body. Hence, we need to have some caution in their use. But class 4, they are size below 100 nanometers and non-biodegradable, wherein there may be an accumulation of these particular nanoparticles in a particular tissue causing a lot of other issues. So the persistency scale goes on increasing with these particular things and size is like this. So it is a nanomaterial classification system proposed by Keck and Muller and now being adopted by different scientists for the purpose. The third part is the pharmacokinetics and dynamics. So pharmacokinetics and distribution of nanoparticles in the body depends on the surface physicochemical characteristics, shape and size of the nanomaterials. So nanomaterials with 10 nanometer size can be preferentially found in blood, liver, spleen, kidney, testis, thymus, heart. You can see all the organs of our body, they can penetrate and they can go into all parts. So the surface of the nanoparticles can also impart upon distribution of these organs, and combination serum proteins may be also seen influencing their cellular uptake. Once these studies are complete, the last problem is scale up and reproducibility. Nanomedicine manufacturing process have 2 different approaches. One is top-down approach, the other one is bottom-up approaches. So both these approaches are going to involve different steps like homogenization, sonication, milling, emulsification, and we may be using different organic solvents, and finally these organic solvents are subjected for evaporation. So the experimental conditions for the production involves the control of various parameters like speed, mixing time, milling time. All these are going to be very, very critical in determining the size, shape, and stability of the nanoparticles. But in small scale, because we are making them under the laboratory or on the pilot scale, we may be having the control of all these parameters. But when we are going for the industrial production, even a slight variation in the process may cause some critical changes in the physicochemical properties, and they may comprise for the quality and safety of the nanomedicines, and it may result even the therapeutic outcomes. So we have to have a technology to identify these critical parameters and to validate these parameters and to stabilize these parameters. So we may have approaches like QBD, quality by design, and US FDA has proposed process analytical technology approach, where the systematic evaluation and control of nanomedicines can be have more control. So what is PAT? PAT is a system for designing, analyzing, and controlling the manufacturing through timely measurements and a critical quality and performance attributes of raw and in-process materials and processes with a goal of ensuring final product quality. So the whole object of PAT is we have to identify, we have to design, we have to analyze these parameters which are going to affect the quality and performance of the nanomaterials, both from the raw material point of view, during the in-process control and final process control. And the ultimate goal is to achieve a final product quality. So by using these PAT tools, we can evaluate the critical quality performance attributes, and we can assure the quality of the manufacturing product. The ethical issues are also very important, because majority of these nanomedicines are prepared for the drugs which are not suitable for regular use, for example, anti-cancer drugs. So by the time these nanomedicines are going to be subjected for human critical trials on patients, they might have received many different treatments without benefit or anything on the particular disease for which this particular nanomedicine is being prepared. Though we may be obtaining a valuable knowledge by these clinical trials, the advantage that is going to be derived by the individual is almost negligible. That means there is no individual benefit that is obtained. But we may say we are getting a good benefit for the total population. We can ignore the benefit to the individual, but at the same time, we need not forget the ethical issue in that. And we have to establish the benefit-harm ratio and the effects that are going to be caused by the nanoformulations, which are unknown to a major extent. So we have to have a clear understanding of this benefit-harm ratio before the clinical trials are taken up, because we have to answer so many questions. Because whenever a protocol is submitted for ethical clearance, the ethical committee members may raise so many concerns which may not be properly answered by the researcher about the risks, benefits, likely outcome of the trials, and other humanitarian issues. So I mean to say there is a gap that is going to exist between the questions raised by these ethical committees and the answers that are being given by the particular researcher, because majority of these questions are not having proper answers. So we need to bridge this gap very carefully. And when once all this is successful, the regulator concern is always related to biosafety. What is the efficacy of the product compared to the conventional dosage form? What about the stability of the product? What about the dose and exposure times? So whenever the nanomedicine is subjected for approval by the regulator, we need to answer all these 4 questions very clearly, and then only it is subjected for any approval. So one need to think about the regulatory aspect also. So when you take the regulatory comparisons of US, European Union, and India, US having 5 groups which are going to control the approval process of this nanomedicine, like Center for Drug Evaluation and Research, Center for Devices and Radiological Health, Center for Biologics Evaluation and Research, Nanotechnology Interest Group, Nanotechnology Task Force. And in the Union, you have 3 groups, Committee for Medicinal Products Use, Innovation Task Force, and New and Emerging Technologies Working Group. And from Indian point of view, we have Ministry of Health and Family Welfare, Central Drug Standard Control Organization, Indian Pharmacopoeia Commission, National Pharmaceutical Pricing Authority, because finally the price of the product is also very important. And hence India is going to have all these 4 bodies which are going to involve in the approval process. As far as India is concerned, there are no proper guidelines for the evaluation of nanopharmaceuticals till 2019. So they formulated different committees, and finally, in the month October 2019, Government of India has published guidelines for evaluation of nanopharmaceuticals in India. And the safety studies, what is the nano size range, all these are given proper importance while designing these safety guidelines, and they have to have the specific guidelines that are followed as per the second schedule of the new drug clinical trials rules. And the nano size range should be declared on the product specifications, and particles should be in the nano size range in the given testing conditions during the claimed stability period of the product. So when you are going to give the label for this particular nano formulation, this is going to be very, very important and critical. So according to these regulations, the nano formulations are categorized into 4 different categories. Category 1, here the drug is new and the carrier that is being used is also a new carrier. Hence, one should establish the quality, safety, and efficacy as specified in the second schedule of the New Drugs and Clinical Trials rules of 2019. So unless these specifications are properly documented, the approval cannot be given. Category 2, which is almost a very, because it is given a green color, drug is new, but carrier is approved as nanopharmaceutical. So one need to prove the independent studies for the assessment of safety performance of the such carrier may not be required because the carrier is already tested for its safety and efficacy. And category 3, drug is approved, nano carrier is new. So we have to establish the evidence of safety and efficacy of the nano material, and the final product should be provided. And category 4, both the drug and carrier are already approved in other countries, but not in India. So we have to give the abbreviated or bridging studies, whatever that are required by the regulator as per the new drugs and the clinical trials rules of 2019. So we have to specifically address some of these points, like what is the basis for making this particular nanopharmaceutical for the claiming of improved safety, efficacy, and reduction of the toxicity profile, reduction of the dose, whatever that is being claimed as new, one need to establish without any doubt. And any issue that is related to the different pharmacokinetics or pharmacodynamics than that of the conventional drug must be explicitly proved. And the specific adverse effect or property of formulation compared to the traditional drug or the conventional formulation, the effects on vital systems must be properly established before applying for approval of these nanopharmaceuticals under this particular direction. So one need to give the information related to the nanopharmaceutical that is submitted for approval containing the introduction, chemical and pharmaceutical formulation, animal pharmacology studies, animal toxicology studies, human clinical pharmacology phase 1, phase 2, phase 3, and special studies, whatever that are required, and what is the regulatory status of this particular nanopharmaceutical made in India in relation to the approvals that are given for the similar product in other countries. What are the samples and testing protocols that are going to be followed, and what is the information that is going to be provided on the label? Unless this data is provided, no formulation is going to be approved under the nano formulation category by the government of India as per the present guidelines. So what are the future prospects, what we can see? Nanoscale devices can sense minute differences at the level of single molecules and single cells. And we have exquisite sensitivity for single cell heterogeneity and extremely high throughput, revealing hierarchies and subpopulations. And we can have traditional bulk assays for the normal conventional drug, but we can have the specific assays for these particular nano formulations. So we have different approaches using this nanotechnology to cause the perturbance in the cells, and they can be designed to precisely deliver the therapeutics to a targeted location very accurately compared to the conventional dosage forms. So we can avoid certain biological barriers, and we can alter the pharmacokinetics because they are specifically targeted to a particular organ, and we can also change the biodistribution of the particular drug. And it can also avoid the complications associated with the conventional delivery and other uptakes. So we can also apply this nanomedicine for personalized medicine because certain patients require a specific doses. So if one can develop a technology, we can develop a specific dose drug for that particular patient who requires a personalized medicine, and we can diagnose the patient, what is the requirement of the patient, and how it is going to perturb the cells, and we can develop an integrated nanomedicine for achieving this personalized medicine goal. But it is only an assumption, and it is in the experimental stage so far, and in future I think we can see this particular approach. So what I would like to conclude here is, one should be very careful in developing the nanomedicine. First of all, a particular case study must be undertaken for preparing a nanomedicine for the given drug or for preparation of a new carrier system. What are the advantages? What are the disadvantages? And how we are going to target that particular specific site is to be established. Then we can go into the wet lab for preparation of this nanomaterial. And we need to use proper evaluation techniques for the size distribution, data potential, surface characterization, all these things. So majority of these equipment are very costly, and we need to have good laboratory facilities. Then only the work can be taken up. I'm not discouraging the young researchers that in a small college we cannot take up this work, but we need to have a very good collaborative research programs wherein these facilities are available such that the systems can be made in our laboratory, but we can transfer them for evaluation to those particular laboratories where these facilities are well established. And once we are successful, we can scale up process for optimization. And in this process of scale-up optimization, we need to establish the reproducibility of the system at different stages. And we have to carry out the in vivo studies following the ethical guidelines. The proper nanoparticles in the body. And last but not the is important because the regulations are quite different from one region to the region. In interest of marketing, we need to get the approval from these particular region regulators. And last but not the least, in my opinion, this is only my pure opinion. It is not we need to collaborative research. Being pharmacists, we cannot claim that I'm a good nanopharmaceutical scientist because majority of the works, what we have published from our laboratory, they are involving a lot of collaborations. In the first work, my scholar has gone to US for establishing its physicochemical characteristics in one university. And in the second, third, fourth works also, we used a collaborative research wherein we have roped into our research, like scientists from chemistry faculty, scientists from physics faculty, scientists from biomedical engineering faculty, and even from medicine faculty also, we roped in some of these people such that the comprehensive evaluation can be more easy compared to an independent approach, wherein I prepare the nanopharmaceutical, I evaluate the nanopharmaceutical. That means I should be avoided in the nanopharmaceutical research, and we should be always the best proposition for a successful story. So if we can take all these things into our mind, certainly we can develop a good nanopharmaceutical which can be taken from the bench to the market level. And once again, I thank Vishnu College of Pharmacy for giving me this excellent opportunity. I think I rushed up a little bit for some reason, which some of my few friends only know why I rushed through this seminar, because I have something after 10:30 which are very important for me. So please bear with that. And I'm sorry for rushing through a little fast because of the reason. And I thank the audience for their patience. So, because we are in the COVID era, we say thank you and saying namaskaram from our Indian culture. And anybody who has any questions or doubts, they can settle me, mail me, and I'll respond within a day.
Thank you, sir, for your elaborative session and throwing light on the future perspectives and the current trends on the nanotechnology as well as the novel drug delivery systems. Now the session is open for the questions. The people who are in the Zoom can directly ask the question by introducing yourself and from your introducing about your institution. Directly you can pause, you can ask the question, people who are in the Zoom session. And the questions from the YouTube chat box will be made audible to the sir through me. Then you can get the respective answers recording.
Hello.
Yeah.
Sir, my name is Vasu, sir. I am from Sri Vishnu College of Pharmacy.
Okay.
Sir, my doubt is in the last very slide you have told that personalized medicine.
Yeah.
So in that case, what are the challenges for clinical trials? Clinical trials.
See, personalized medicine is always for the established drugs and systems. Only thing is we are going to titrate the dose according to the need of the patient. So in that case, we need not go for a clinical trial elaborately as that of the regular dosage form development. That's my personal opinion.
Thank you, sir. Thank you, sir.
Sir, it is a question from Mr. Prasad, Koringa College of Pharmacy, Kakinada.
Yeah.
He would like to ask, apart from the size characterization for the dendrimers, any other specific test, specific evaluation tests?
We need to establish its generation by using the IR, NMR, and all these studies. Because what is the generation, fourth generation, or third generation, or second generation? That is also very essential. When you synthesize a dendrimer, if you take a dendrimer, there is already established in the market, there is no need.
Okay. Sir, question
Yeah, Ashok, anything else?
Okay, sir. Further questions will be answered through the email link provided to them. We will provide the email link in the YouTube channel, such that any person in specific questions, they will be directed to you directly, sir.
Thank you very much.
And thank you for an enlightening session. Now I request our Prasad sir to give the concluding remarks regarding the today's session. Over to Prasad sir, Principal sir.
So, good afternoon, sir. Hello, can you hear me?
Yeah, Prasad, we are able to listen.
So, on behalf of Sri Vishnu Education Society and on behalf of Sri Vishnu College of Pharmacy, I am very glad to say the First of all, I congratulate Dr. Ramamurthy sir immediately accepting our invitation in spite of his busy schedule and by giving us such a beautiful lecture and enlightening about the nanotechnology information. And then, so sir, thank you for your active participation and immediate response from our side when you are asked. Thank you for coming there. We expect this similar type of participation from you in the future activities. Once again, I thank all the participants and I request the participants to completely participate in all the 5 days and then enthusiastically involved by asking questions and then by updating the knowledge regarding the topics related to the most of the topics. In a way, even though they are connected to the nanotechnology, most of them are very advanced topics what are going to. Thank you. Thank you for.
Thank you, everyone.
Okay, thank you, sir.
మీటింగ్ అనేది కాదనుకుంటారు వేరే వాళ్ళది నేను పైన పెట్టిన దానికి ఏమో అనుకుంటున్నా ఐపీఎస్
రైట్ సార్ అయిపోయిందా అది క్లోజ్ చేస్తాను సార్ ఇట్లా
Yeah, thank you, Ashok. Everybody.
Yeah, Ashok, it is over to you. So would you like to close the session?
Okay, and I would like thank you to one and all for your extended cooperation and making this today's day 1 event a grand success. Now I would like to declare the session is closed. The feedback link is provided at the YouTube channel and it will be available up to 12 hours after the session. Please do the feedback and submit, then only you are eligible to get the certificates. This is the information regarding to all the participants and the delegates. Thank you.
Yeah, so thanks a lot, sir. I know why you are rushing, and in spite of that challenge, you have accepted and delivered the talk very fluently and very extensively. I'm really impressed, and as always, with your talks and your presentation. I'm sure all the participants are really enjoyed your session and wish you good time ahead for today and
Thank you.
the next thing you're going at. Yeah, thank you. So thank you all. So we are closing the session now.